then I think we're ready for our final speaker, which is Ansuman Satpathy. Ansu is an assistant professor in the Department of Pathology at Stanford, and he's a member of the Stanford Cancer Institute, the Parker Institute for Cancer Immunotherapy, and the Immunology, Cancer Biology, and Biomedical Informatics Programs, BioX, and a faculty fellow in ChEM-H. He is a major collaborator for a lot of our institute members, which is why he's speaking here, and his research group focuses on developing and applying genome-scale technologies to study fundamental properties of the immune system in health, infection, and cancer. Thanks, Ansu.
Thank you, Stacy, and thanks to Alex, Stacy, and the rest of the organizers. Really a pleasure to be part of this discussion and symposium today. So as Stacy mentioned, my lab works on high-throughput genomics, and I will touch upon some of the work that we've done over the past 2 or 3 years, trying to apply genomics to understand cancer immunology. So broadly, my group is interested in developing and applying novel high-throughput technologies, in particular genome sequencing technologies, to understand primary immune cells in patients, and particularly in patients with cancer that are receiving immunotherapy. And we think about this at many levels and scales, and we think about using single-cell genomics to understand what are the constituent cell types in the tumor microenvironment, which of those are responding to immunotherapies, which of them are beneficial or not. We think about pairing that information at the single cell level with orthogonal information about the cell, things like TCR sequencing that allows us to pair phenotype with antigen specificity or clonal lineage tracing. We think a lot about gene regulation, in particular understanding epigenomics and how certain specific cell states are established and maintained. And then we try and use all of this information to really understand the wiring of immune cells and how we could potentially use genome engineering technologies to improve the function of immunotherapies in patients. And then finally, and I'll just touch on this at the end, we not only think about engineering immune cells, but we also think about how do you properly target those immune cells, engineered or not, to the right antigens to effectively but safely target cancer cells. And today I'll try and touch on sort of each of these topics very briefly to give you a sense of all of these aspects of our work. So I don't think I need to repeat this after really a huge number of wonderful talks today. But suffice to say that over the last 10 years, we've really seen a transformation in how we treat cancer in patients in the clinic. And this is really built on the foundational work of Jim Allison and Tasuku Hanjo, who discovered inhibitory receptors on T cells that could be blocked to induce or reinvigorate immune response against cancer in a patient. And so now for many patients in cancer types, we, instead of giving radiation or chemotherapy as first-line therapy, we now give agents that activate the patient's own immune system to kill the cancer. And here's an example of a patient very early who received anti-CTLA-4 checkpoint blockade, had metastatic disease in the chest cavity, and quickly went away after that therapy and remained sort of disease-free for many years after. So I think to me, this really shows the promise of immunotherapy. When it works, it really works. Unfortunately, what we now know is that for many patients, it doesn't work well. So in many cancer types, and even patients who have cancer that theoretically should work, should respond to immunotherapy, you don't get a response. About 60 to 70% of patients don't respond. And so that's really the driving force of what our lab is trying to understand. Why does it work when it works? What are the fundamental mechanisms of cancer immunity and immunotherapy? And why does it not work in some patients? And how can we really understand the biology of immunotherapies to engineer a novel or combination of therapies that really help patients? And the way that we really got into this was through a collaboration with Ann and Howard Chang at Stanford, studying patients with basal cell carcinoma. So as many of you know, this is quite a common cancer in the US, about 2 million new cases a year. And unfortunately, when this cancer advances and metastasizes, treatment options for these patients are quite limited. And so Ann's group did one of the first monotherapy PD-1 trials in this patient population, patients with advanced BCC. and showed that we could really get a really good response rate. About half of patients derived some clinical benefit. Some of those patients went on to have sort of durable responses for many years now. The benefit of studying patients with BCC is that the tumors are quite large and they're superficial, they're on the skin. And so you can get samples from these patients both pre and post therapy, and that really allows you to ask some very key questions, such as what are the diversity of the T cell phenotypes in the tumor microenvironment? Is it just one T cell type? Is it many? Which of them actually are responding to the immunotherapy, to PD-1 blockade? And importantly, where are these cells coming from? Are they and the sort of the common view in the field at the time was that the way that checkpoint blockade, in particular PD-1 blockade, works is by reinvigorating cells that were already in the tumor microenvironment. But to really understand this, you need to have a clonal assay that can measure and track individual T cells in the same tumor in the same patient pre and post therapy. So that's what we did. We used patients that were in this trial. And isolated samples from 11 patients, both pre and post therapy, site matched at the same place. And so we isolated the tumors, dissociated into single cells, and then performed single cell paired RNA-seq and TCR alpha-beta sequencing in high throughput. And so what I'm showing you on the bottom here is a reduced dimensionality plot. On the left is the phenotypes of the cells, and on the right is the TCR sequence. And at the time, we were quite surprised to see really the vast diversity of T cell types that exist in the tumor microenvironment in patients. Some of us had assumed that it would only be CD8 T cells, maybe there would be some Tregs in there, but you really see the full diversity of CD4 T cell types, including Th17 cells, TF8 cells, even some naive cells. And then on the right, you see really the full differentiation spectrum of CD8 T cells as they become activated, exhausted through chronic stimulation, and then also as some of them form memory. And on the right, it's the same UMAP, but now colored by whether we were able to obtain paired alpha-beta TCR sequencing. So if we did, the cell is colored blue. If we didn't, it's gray. And you can see that most of the map is colored blue. And so what this really allowed us to do, as many of you know, the TCR is somatically recombined during T cell development. And so now we can actually track the fate of individual T cells in patients over time, since these TCRs are inherited in daughter cells from parent cells because they're somatically recombined. So what does this data look like? And the first question that we asked was, what population or how many or what fraction of the T cells in the tumor microenvironment were actually responding to PD-1 blockade? And we could use clonal expansion and Gini index of the TCR sequences as a surrogate measure for this. And so what I'm showing on the left is clonal expansion of various CD8 T cell types, pre and post therapy, colored in different colors. And so what you can see is that really only one population of CD8 T cells clonally expanded post-therapy, and that was this cell type that's classified by T cell exhaustion that we've heard about already today. And really this made up about 2% or so of all of the T cells in the tumor microenvironment. So really going against the idea that PD-1 blockade is affecting a large number of T cells, and actually most of the cells in tumor are bystanders to this therapy. We could then layer on the transcriptional data from single cells onto this clonal expansion landscape and start to link together a couple of different features of responding T cells. So first, these exhausted T cells, as I said, were clonally expanded. They also expressed markers of tumor specificity, including ENT-PD1 or CD39 that was demonstrated by Evan Newell's group. And they also had features of T cell exhaustion. So we're now starting to link together using this multimodal genomic assays the idea that T cells are clonally expanding, are recognizing the tumor, and are becoming exhausted over time by seeing this chronic antigen in the tumor. And then next, we asked sort of the question that I set up in the beginning, which is that of these cells that are responding to PD-1 and clonally expanding post-therapy, where are they coming from? How many of them were in the tumor in the same patient at the same site pre-therapy? And so what I'm showing you on the left is the fraction of the expanded clones in each category of T cell that was novel, or in other words, not seen pre-therapy in the same tumor. And what you can appreciate is that the vast majority of responding clonally expanding cells post-therapy were not present in that patient, in that tumor pre-therapy, or at least not clonally expanded. So what this tells us is that the way, or at least a way, that checkpoint blockade works is not by simply reinvigorating T cells that are already in the tumor microenvironment, but by recruiting a novel set of T cell clones or new army of T cell clonotypes to fight the cancer. So why is that the case? Why were the cells that were tumor-specific and exhausted pre-therapy not reinvigorated and not induced to clonally expand after PD-1 blockade? Was there something about the molecular state of that cell that made it sort of dysfunctional in response to PD-1 blockade? And so to answer this question, we developed a method called single-cell Ataxia, or assay for transposase accessible chromatin with sequencing. That allows you to understand the open chromatin landscape and generally the epigenomic state of a cell. And what we wanted to do was adapt this assay to be one, high throughput, and two, to be amenable to primary human immune cells. And so we worked together with Grace Zhang and the 10x Genomics team, really led by the work of a graduate student, Jeff Granja, to develop and adapt their 10x single-cell droplet system to perform Ataxia. I won't go through the details of the method for the sake of time, but the general idea is that we're able to transpose cells and perform Ataxia in bulk while maintaining the nuclear membrane as the cell partition. And then we load these transposed cells into droplets and then perform standard molecular biology to amplify open chromatin sites. So this is the data. This is now showing you a UMAP of T cells in the same BCC patients, actually exactly the same samples that we did single cell RNA and TCR on, but now the UMAP is representative of every dot, every cell's epigenetic state, so open chromatin sites. I don't have to take you through the T cells, exactly the same subtypes, so CD4 cells on the left and CD8 on the right. But what we found was that exhausted T cells actually exhibited quite a distinct epigenetic state, just as distinct as any other T cell type that we're accustomed to thinking about, Th17 cells, Tregs, et cetera. And so there was a thought in the field that T cell exhaustion might just represent the overexpression or new expression of inhibitory receptors on the surface of the cell. And that's really all that was blocking the cell from being reinvigorated by PD-1 blockade or other measures. But what we're showing is that T cell exhaustion, and this has been shown by several other groups in other settings, John Wary, Andrea Scheedinger, Nick Haining, and many others, that T cell exhaustion is really accompanied by whole-scale epigenetic remodeling, and that's likely the reason that these cells are not functioning the same as effector T cells or other cells. And importantly, what happened after PD-1 blockade? Was it true that the epigenetic state of T cell exhaustion was not reversed and was stable? And so what I'm showing you here is the same UMAP of single-cell Ataxia data from T cells in the tumor, but now colored by whether that cell was obtained from a pre or post-therapy sample. And so what you can see is that this exhaustion cluster is exactly the same post-therapy. So PD-1 blockade does not change the epigenetic state of exhausted T cells, and even the new clones that are coming in that are recruited into the tumor still eventually enter this exhaustion pathway. So what I've shown you in this first part is that a large number of clonally expanded T cells post-therapy are not found in the same tumor pre-therapy. And so what this means is that PD-1 blockade does not reinvigorate exhausted T cells in the tumor, but likely works at least in an additive fashion by expanding novel T cell clones and recruiting them to the tumor microenvironment. This is likely due to a distinct epigenetic state and lineage commitment that happens in T cell exhaustion. And I think the point is that what this data is telling us is that this epigenetic state of exhaustion is not reversed by checkpoint blockade and represents a significant barrier to maintaining a long-term T cell response in cancer immunotherapy. So having seen that, what a lot of my lab thinks about is, is there a way and can we reverse or prevent T cell exhaustion to extend the lifespan of a T cell's response to cancer? And before we can really answer that question, the first thing we should ask is, do all T cell clones become exhausted in response to seeing a chronic antigen stimulation like a tumor? Or has the body evolved ways to actually handle this by itself, right? So are there clones that see chronic antigen in a tumor or chronic viral infection or any other setting that in fact don't become terminally exhausted? And can we learn from the molecular wiring of those cells? So to answer this question, we generated a large genomic atlas of T-cell exhaustion. In this case, in the setting of sort of the classic model of T-cell exhaustion of chronic LCMV infection. And so just to orient you to the data, so we've generated single-cell RNA and TCR sequencing data, also single-cell ataxic data, and we'll just focus on this plot here on the left. And so what you're seeing is sort of the developmental stages of T-cell exhaustion that occurs in chronic infection versus what happens as the cells go sort of down in this UMAP in the setting of acute LCMV infection. So this is an infection setting that doesn't cause exhaustion. And what we see, and this is again similar to what many others are describing now in the field, is several stages of the development of T-cell exhaustion seen both at the transcriptional level and at the epigenetic level. It starts with this progenitor exhausted phenotype. So these are cells that express high levels of TCF7, sort of stem-like, if you want to think about it that way, and reside often in lymphoid organs and can lead to a persistent T-cell response and the generation of more and more exhausted T-cells downstream. You see this intermediate cell state, which are called transitory exhausted cells. These actually have a lot of effector function, sort of classified by their expression of CX3CR1 and also PD1. And then you see development of terminal exhaustion. This is sort of classically how you think of exhaustion, a dysfunctional cell with really high levels of all inhibitory receptors and doesn't proliferate or secrete cytokines. So as a cell gets chronic stimulation, it sort of traverses this path going from a progenitor to a transitory to a terminal exhausted cell. And you can see different flavors of terminal exhaustion depending on the organ that you look at. In contrast, what we see in acute infection is exactly what you would expect. You get the development of an effector T cell, and then after the infection resolves, you get development of T cell memory. Since we had paired TCR data, again, we could ask what's actually happening to at a clonal level to every single TCR clone across thousands and thousands of clones in a polyclonal response. Is it that every clone goes down exactly the same terminal exhaustion pathway, or are there other phenotypic outcomes? And so to orient you, it's essentially the same plot, just sort of flipped around. Acute infection is going up, terminal exhaustion is going down in chronic infection. And now what you're looking at in these plots on the top right here are clonal expansion data. So in acute infection at day 8, the blue cells are the clonally expanded cells in these mice. And these are, as we expect, effector cells. At day 21, the clonally expanded cells are all some form of memory cell. What you see in chronic infection and development of T-cell exhaustion is something quite different. Already at day 8, these cells look very different. And at day 21, most of them are going down this terminal exhaustion pathway. You do see some cells in effector memory. We'll come back to that. Now what's happening at the level of individual clones? So what these plots are showing you is every bar, every column is an individual TCR clone with a unique TCR sequence. The height of the bar represents clone size, and the coloring represents which of those cells are in an individual phenotype. And so again, in acute infection, early, it's all effector cells. Late, it's all some flavor of memory cell. In chronic infection, these are just the top 10 expanded clones. You see most of the cells in either progenitor or transitory early exhaustion phenotype. That's probably what we would expect. At late time points, you see the development of terminal exhaustion. But what we were surprised to see was a set of clones that were clonally expanded. They were antigen specific. We could determine that from tetramer staining. But they were not going down this sort of classic path of terminal exhaustion. They were instead becoming this effector memory-like cell. So what that tells us is that there is a subset of cells and subset of TCR clonotypes that in a polyclonal response see chronic antigen stimulation but don't become exhausted. And so if we zoom out and sort of sum this up across all of the clones that we see across all of the mice, what we see again is this is the classic view of T-cell exhaustion, and about half of the clones go down that classic path. And then there are two alternative paths that lead to the development of effector memory cells, either exclusively or in addition to terminally exhausted cells. So why is that, and what can we learn from that? And so we've been trying to understand what is it that's different about these cells that go down the effector memory path. And one hint is that if we sort out TCR clones that are low affinity TCRs. As a surrogate, we're using low tetramer staining. We get a huge enrichment in the development of effector memory cells. So this tells us that what's determining, at least in part, the phenotypic outcome of what happens in the setting of chronic antigen stimulation is the strength of TCR signaling itself. So high affinity clones go down this terminal exhaustion path, low affinity clones preferentially for memory. And you can envision why evolutionarily that would be an advantage of a polyclonal T cell response. Okay, so what I've told you is that the TCR signal strength itself can lead to increased persistence and effector memory formation in T cells that see chronic antigen. But can we actually genetically determine that path? So instead of just putting a strong or a weak TCR on the cell, can we figure out sort of the molecular levels that would allow us to manipulate whether a cell goes down one path or another? And why might this be useful? And I think, given the set of talks today, I don't think I have to belabor this point. But as we know, chimeric antigen receptor therapies utilize sort of a similar TCR signaling pathways. And importantly, what we showed together with Crystal Makel's lab and Evan Weber, who's on the one of the listeners here, is that CAR T cells also get exhausted, and that exhaustion looks very similar transcriptionally and epigenetically to what I've already described to you in the context of endogenous T cells. And then the final sort of piece of evidence is what Carl has already presented, and we were quite fortunate to tag along to really Ed Stadmauer and Carl June's tremendous effort to do the first in human CRISPR engineering trial in cell therapies. And the take-home point is that what you're able to do now is edit T cells, and it seems to be safe and effective. And so all of this sets up the idea that not only is exhaustion important in the setting of endogenous T cell responses and checkpoint blockade, but also in the setting of cellular therapy. And so if we had a way to manipulate the persistence genetically, that might actually be actionable in a patient therapy. And so how do we do this? And so we took two strategies. So one is using in vitro model, using chronic activation, antigen activation in a dish. And a second is to use a model of in vivo tumor infiltration and persistence. And so what we're doing is performing genome-wide CRISPR screens to identify factors that if you delete them would persist longer in the setting of chronic antigen stimulation. This is really the hard work of Julia, Winnie, and me in the lab. And I'll just pause for a second to tell you that we've heard a lot about the chronic antigen stimulation systems. At an epigenetic level, what you get in chronic antigen stimulation alone, so if you just put T cells in addition, repetitively stimulate them with antigen or with CDR3 stimulation, what happens to the cell epigenetically looks very similar to what happens in the context of an endogenous T cell. So again, I'm telling you that a cell therapy, exhaustion cell therapy is potentially quite similar to what happens in TILs, and that most of that epigenetic landscape and dysfunctional landscape is set up by the chronic stimulation through the TCR itself, and not sort of extraneous tumor microenvironment factors. Of course, those can, in addition, induce dysfunctional signatures in T cells, but we think that the chronic TCR stimulation is really the main thing that's driving at least the exhaustion phenotype. Okay, so this is the results of the screen. So this is the result of the in vitro screen, where again, we've performed a genome-wide CRISPR screen to identify factors that improve persistence. In this setting, positive controls for the screen are TCR signaling factors. So again, what we're doing is just giving chronic TCR stimulation in a dish. So if you knock out TCR, you can the cells will survive longer because you're not inducing dysfunction and death. So this serves as a positive control that the entire screen worked. In addition to TCR signaling hits, you can see a number of additional functional categories, including cell adhesion receptors, metabolic enzymes and mitochondrial factors, and also a large set of epigenetic factors, including histone modifiers, INO80 complex and SWI/SNF members, and some transcription factors. And I'll just point out that inhibitory receptor deletion, including PD-1 and CTLA-4, actually were worse for the cell, so they did not improve persistence. If we now compare the hits in the screen in vitro to what happens in vivo, I'm now coloring the same protein-protein interaction heat map with the factors that validated or were also good for T cell persistence in vivo. And so if they were good, deletion of the factor was good for the cell, it's colored red. If it was bad, it's colored blue. And so what you can see is that we get a huge enrichment of factors that are classified as epigenetic factors, suggesting that perturbation of epigenetic modifiers improves persistence of T cells in the face of exhaustion and chronic antigen. So why might this be? And we're still trying to understand this, but one way in which we've tried to understand it is by performing in vivo perturb-seq experiments. So what we're doing is introducing our guides into T cells, transplanting them into mice, isolating the T cells out of the tumor microenvironment, and then performing single-cell RNA-seq paired with the guide identity that cell receives, so the perturbation identity. And so here are the phenotypes of the cells that come out of the TILs, just based on RNA-seq. And you can see a relatively similar progression of exhaustion. So you see this TCF7 progenitor exhausted cell, see an intermediate cell, and then you see a terminally exhausted cell. And what we found is that perturbation of a number of these epigenetic factors had a common effect on the distribution of phenotypes of the cells across this sort of differentiation trajectory of exhaustion. And that is that they were enriched in the early progenitor state and sort of depleted in terminal exhaustion state. So what this suggests is that depletion of epigenetic factors somehow blocks the sort of business end of the strong TCR signaling and maintains cells in an earlier progenitor state rather than allowing them to progress down terminal exhaustion. And that's good for maintaining persistence of T cells. So what I've shown you in the second part is that T cells can avoid exhaustion. If you look at a polyclonal response to chronic antigen by tracking many clones, what you see is divergent differentiation trajectories, and not every T cell clone that sees chronic antigen becomes terminally exhausted. We performed genome-wide CRISPR screens and identified several epigenetic factors as novel targets to disrupt and improve T cell persistence. And we think this works by increasing the frequency of cells or maintaining the cells for a longer time in this progenitor exhausted state. And finally, I just want to switch to the other side of the coin, which is that we not only think about engineering T cells and understanding what's happening sort of cell intrinsically in a T cell in the context of solid tumors or cancer immunotherapy, but also the other side. How do we direct the T cell to the right antigen? And is there something to be done to use high throughput genomics to identify the antigens in a more precise and sort of more unbiased way? And so we really came to this after we saw the results of a couple of CD19 CAR T studies published in New England Journal of Medicine, including from Carl, which showed tremendous efficacy of the therapy in patients, but really a high incidence of neurotoxicity. And in particular, we are struck by the fact that CARs or bispecific T cell engagers or other antigen-directed therapies that target CD19 had a higher incidence compared to therapies that targeted other B cell antigens, CD20, CD22, in often the same patient population. So it really seemed like there was something about CD19 that was increasing this incidence. And so at the same time, we and many people here have been involved in sort of generating this human cell atlas, right? So we believe that if we generate a sort of compendium of all of the cell types and all of the gene expression across healthy and diseased individuals at the single cell level across multiple omic spaces, that will allow us to understand disease processes. But perhaps this could also allow us to understand antigen expression at a depth that we hadn't before. And so combining these two ideas, what we did is looked for, relatively simple idea, looked for the expression of CD19 in human brain. Was there a cell type in the human brain that expressed CD19 that we had previously missed? And so a lot of this data is publicly available. And so what we did is download a bunch of human brain single cell RNA-seq data sets. I'm showing you the results from one of those here. And looked at the non-neuronal cells in the brain. And so I can tell you that neurons don't express CD19, at least not in data that we've seen. But in non-neuronal cells, we could segregate these into various sort of glial cell types and also some immune cells. And we could ask, are there cells that express CD19 that are not B cells and are not cancer cells? And so indeed, we found a rare population of cells called Mural cells or pericytes that express high levels of CD19, but not CD79 or other B cell antigens. So what are Mural cells? And a lot of work has been done by our colleagues here at Stanford, Ben Beres and others, showing that if you deplete pericytes or Mural cells in mice, that you get vascular leakage. And the function of these cells is to tightly wrap around endothelial cells surrounding blood vessels and maintain the integrity of tight junctions between endothelial cells. So to maintain blood-brain barrier integrity. So the question is, if that's the case, then as I said in the beginning, this should be a CD19 specific expression and not seen in 20 and 22. And so what we did is collaborate with Arnold Krigstein and Aparna at UCSF to look at a really large data set of about 1 million single cells of Mural cells as they were developing in the human brain. And just to make a long story short, as cells go from yellow to dark blue, they're developing into Mural cells in the brain, and the blue cells are sort of the earliest emergence of this lineage. And so what you can see is that CD19 is expressed across multiple Mural cell subtypes, so pericytes, as I mentioned, but also vascular smooth muscle cells, which surround larger vessels in the brain. And this is really not the case for CD20 or CD22. So all of this starts to fit together, that a unique 19 expression, and it's only on in Mural cells in the brain and not other organs. And depletion of those cells might lead to vascular permeability that we see in patients receiving these CARs. And I think Carl already showed this data, but we collaborated with Carl, Avery Posey, and Dennis at UPenn to show that transplant or infusion of murine CD19 recognizing CAR T cells in mice that lacked B cells could lead to vascular permeability, suggesting that there was a non-B cell target that could lead to neurotox. And I think the real point here is that this is not, or at least we believe this is not only a CD19 problem. So we've looked across a number of antigens that are used in CAR T or BiTE or ADC therapy currently in the clinic or currently in trials. And we see off-target expression on healthy cells in the human cell atlas in almost every case. So ROR1 is expressed in the lung, CD33, and some non-malignant hematopoietic cells, obviously. BCMA is actually expressed in the brain, and several others. And so I think the take-home point from this part of the talk is what's bolded here, which is that what we've shown you is that CD19 is expressed in a rare population of brain pericytes, and maybe this contributes to neurotoxicity. And I think there's a lot to be done in terms of understanding the synergy of pericyte CD19 with CRS and how much each contributes to neurotoxin in each patient. But I think the point is that we should be using these human cell atlas efforts in large-scale genomic and single-cell analysis to really understand, one, what is the target risk of a particular antigen that we're taking forward into patients as a target for cell therapies or others, other antigen-directed therapies. And then second, that we should use that same database of information and genomics to nominate in an unbiased way what would be the best antigens to target in a particular cancer type. And I really think that's a way that the field could move forward. So thank you for your attention. What I tried to give you a sense of is all of the ways in that we're thinking about and applying high-throughput genomics to understand cancer immunotherapy. A lot of this in primary patients, and really trying to go the full spectrum of understanding fundamental aspects of immunology and cancer immunology, and then how we can use that to really engineer better therapies in the future to benefit patients. So thanks for your attention. I'll just end by thanking really all of the wonderful people in my lab who have contributed to the work and wonderful collaborators. I talked about unpublished work from Julia, Bence, Katie, Nee, and Winnie today, and really have had wonderful collaborations, Stanford, UCSF, looking forward to working with the Gladstone Institute more and more moving forward.